Okay, so this is the part 2 of, or actually part 3 of the digestive system. Second week though. So the pancreas we've talked about before in the endocrine system because it's an endocrine gland. Remember, it secretes insulin and glucagon to help regulate blood glucose. It's also an exocrine gland for the digestive system. Its exocrine function is to secrete something called pancreatic juice. Okay, most of the pancreas is made up by these cells called acinar cells. And they release the pancreatic juice into the tiny little ducts, which lead to the pancreatic duct. So this is that pancreatic duct. The pancreatic duct and the common bile duct join. So here's the bile duct and the pancreatic duct at the hematopancreatic ampulla. So this is that ampulla. It's the release of those two products is controlled by the hematopancreatic sphincter. So it surrounds that ampulla and it controls the movement of bile and pancreatic juice in the duodenum. All right. Pancreatic juice has enzymes that digest carbohydrates and nucleic acids, proteins, and then fats too. So here are some of those enzymes. Pancreatic amylase is going to split starch and glycogen into disaccharides. That's just like that salivary amylase we talked about in the mouth. Pancreatic lipase is going to breaks fats down or lipids down into triglycerides. Trypsin digests proteins, and it's released as inactive trypsinogen, and then trypsinogen is activated by that enterokinases we talked about before in the small intestine. So trypsin is going to be inactive trypsinogen, which is then activated by enterokinases in the small intestine. Chymotrypsin is activated by trypsin, and so is carboxypeptidase, which is an enzyme activated by trypsin. So when trypsin becomes active, it activates these two. Both of these digest proteins. Nucleases break down nucleic acids, so DNA and RNA that we take in. Bicarbonate ions are there specifically to raise the pH of the chyme that's coming into the small intestine from the stomach. All right, regulation of pancreatic secretion. Hormones help to regulate that. First one, secretin, stimulates the pancreas to release pancreatic juice that has lots of bicarbonate ions. So secretin, the pancreatic juice has lots of bicarbonate ions. And then cholecystokinin, that CCK we talked about in the stomach, also stimulates the pancreas to release pancreatic juice. But for that one, the juice has higher concentrations of enzymes. Okay, liver structure. Liver's the largest internal organ. It's in the upper right abdominal quadrant, just under the diaphragm. Liver has a lot of blood. There's 4 lobes to the liver. The right lobe is the largest one. This one. Left lobe is smaller. Quadrate lobe is this one up here near the gallbladder, and the caudate lobe is down here by the inferior vena cava. Liver, the lobes themselves are divided into these hepatic lobules. So the hepatic lobules, like this would be a hepatic lobule, are the functional units. Sorry, the lobules are the functional unit of the liver. Lobules have plates of hepatic cells that come out from a central vein. So here you have the central vein and all these plates of hepatic cells. Between the hepatic cells, you have the sinusoids, which come around the plates. Okay, so a couple of things here. We get oxygen poor blood or deoxygenated blood from the digestive tract, goes through the liver in the hepatic portal vein, or branches of it at least. It mixes with this oxygen-rich blood from the hepatic artery, and the blood filters through these sinusoids. So it just kind of flows through these sinusoids. The hepatocytes are there to adjust the nutrient content, or at least the concentrations, as material passes through there. And then the bile that's produced by the hepatocytes leaves the liver through the bile ducts. So here are the bile ducts, and these hepatocytes are going to produce the bile produced there. When we talked about the circulatory system, we talked about how the blood comes through the liver, but here are some of the functions of the liver, and I'm not going to read through all of them, just kind of highlight some of them. First of all, you get glycogen is produced from glucose, but the liver can also break down liver glycogen into glucose. Let's see. Oh, synthesizes plasma proteins we've talked about. Phagocytizes damaged red blood cells, so you get a lot of breakdown of red blood cells here. And then here's another important one. Removes toxins like alcohol and drugs from the blood. And its digestive role is just to secrete bile. Bile is what the hepatic cells produce, and it's mainly made of water. You got these bile salts. The bile salts are there to emulsify fat. So think about that Dawn commercial when they have like greasy water and they dump Dawn, the dish detergent, into the greasy water, and how it all spreads out and you get like little bubbles of fat. That's fat emulsification. The bile pigments, remember, come from the breakdown of old red blood cells, bilirubin and biliverdin, and they are excreted in our feces. Okay, gallbladder. Gallbladder is a place where we store and concentrate the bile. So the cystic duct from the gallbladder joins with a common hepatic duct from the liver to form the common bile duct. So a lot of that bile that's being formed is stored here and then concentrated here. And the bile duct eventually, or the common bile duct, I should say, empties in the duodenum via that hepatopancreatic ampulla that we looked at a few slides ago. And remember, the hepatopancreatic sphincter regulates that movement of bile into the duodenum or duodenum. CCK cholecystokinin, is what causes the gallbladder to contract. It contracts, CCK is released when fats enter the duodenum, and then the bile is released into it. So that's a good reason to release bile when you have fats in there, because then the fats become emulsified in those bile salts. And this slide is just a good wrap-up slide, summary slide for those hormones of the digestive tract. Look what an important role that cholecystokinin plays. All right, parts of the small intestine. Starts with the duodenum, duodenum. I don't care how you say it. You're never going to say it to me anyway. Then that's a smaller part that connects the stomach to the jejunum. Say the jejunum like that, though. The jejunum is a longer piece, and it ends with the ileum. Notice the ileum has a much different spelling than the ileum in the bone. And the ileum leads into the large intestine. So small intestine starts from that pyloric sphincter, and it goes all the way to the beginning of the large intestine. All right. The main function of the small intestine is just to absorb the material that are products of digestion, and then transport any of the waste to the large intestine. Mesentery and greater omentum. The jejunum and ileum are suspended, I guess it's the best word, from this double fold of the peritoneum called the mesentery. Okay, so here's this mesentery. So that's how those are held in there. Part of the mesentery, or part of the function of mesentery, other than suspending those two, is it transports blood and lymphatic vessels and nerves to the wall of the small intestine. And then this double fold of the peritoneum is called the greater omentum. And it kind of falls down from the stomach, where it's attached, over the large intestine and the folds of the small intestine. The structure of the small intestinal wall, kind of the most notable thing, which is kind of cool, is notice that the small intestine has all of these villi that are these intestinal villi, and the purpose of these are to increase surface area. Because if you just had a straight shot across, you wouldn't have as much surface area. You want to have as much surface area in the small intestine as you can, so that you can get more absorption. Because if this was straight across, this area would only be absorbed what was in here. Each villus is made up of these simple columnar cells, and that makes these tougher than, say, simple squamous, but it still allows you to absorb material through there. So the simple columnar cells are there. The lymphatic capillaries and the blood vessels go into the villi to allow you to, remember, the lymph system is where you're going to pull in excess fluid, but also lipids are going to come into here through those lymphatic capillaries called lacteals. The other thing is, if you have some bacteria that you took in with your food, it could also enter the lymphatic system there, so it can be identified and eliminated. So, and there's lots of these goblet cells for mucus. So what we're going to look at now is one of these simple columnar cells. So pull out like this one right here. And this is that simple columnar cell. And notice that the simple columnar cell also has microvilli on it. So this cell makes up the villi, and it has its own microvilli on it to increase the surface area even more, which is kind of cool. The plica circularis are just these circular folds within the small intestine that circular flows of the mucosa, and they also help to increase the surface area. So secretions of the small intestine, well, you get the Brunner's glands. We already talked about those goblet cells secreting out the mucus. The Brunner's glands secrete a mucus, but it's more alkaline, so it has a higher pH. You've got also have intestinal glands, and they're just called intestinal glands, and they secrete a watery fluid. And the watery fluid is there to pick up those products of digestion and bring them into the villi. So there's no enzymes in the intestinal, that watery fluid from the intestinal glands. I kind of feel like people who were naming this stuff just kind of ran out of names when they got, because they were just like intestinal glands, and we're just going to call the fluid a watery fluid. Enzymes in there found in the microvilli. You got peptidases, and they break down peptides into amino acids, so we can absorb amino acids. Sucrase breaks down sucrose, so a disaccharide. Maltase breaks down maltose, a disaccharide. Lactase breaks down lactose, a disaccharide. So all those enzymes are there to break down disaccharides into monosaccharides so they can be absorbed. And then lipase is there to break lipids down into glycerol and fatty acids. Regulation of the small intestinal secretions. Mucus secretion occurs in response to any mechanical stimulation. So the mucus gets secreted anytime there's any stretch in there, or if there's low pH, like if some gastric juice is in there and it has a lower pH. Anytime chyme contacts those goblet cells, they secrete mucus, and intestinal glands secrete watery fluid. When the intestine starts to fill up, it distends the walls, so it expands the walls, and that activates those nerve plexuses on the walls of the small intestine. And that can stimulate those parasympathetic reflexes that cause the release of the enzyme. Or enzymes, I should say. Absorption in the small intestine. The villi are there to increase that surface area. Very little of the material in the small intestine that can be absorbed is not absorbed. So the small intestine absorbs almost everything that it can use, our body can use. All right, so just go through these again. This is a little bit of a review. So carbohydrate digestion and absorption. Remember, we start to digest carbohydrates in our mouth with that salivary amylase. And then the pancreatic amylase continues that digestion as it breaks that starch and those glycogens up into disaccharides. And then those intestinal enzymes, like for example maltose, this disaccharide is broken up by the enzyme maltase into 2 monomers of glucose. And then those monosaccharides just get absorbed via active transport or facilitated diffusion into the villi and then into the blood vessels. Protein digestion and absorption. Well, protein digestion, remember, starts with pepsin in the stomach. It's breaking down the proteins and those polypeptides. And you've got the pancreatic proteases we talked about. For example, there was trypsin, chymotrypsin, break the proteins down into polypeptides, or break the proteins and polypeptides down into smaller peptides. And then the intestinal peptidases break the proteins, or the peptides, I should say, into amino acids. Because we don't want to absorb full proteins because our body can't use those as building blocks. It needs the monomers. So amino acids are absorbed by active transport also into the blood vessels in the villi. Fat absorption is a little more interesting, I guess. The fats are emulsified by the bile salts, remember? So the bile salts kind of split up the fats, and then they get digested by those enzymes in the pancreas, all those lipases in the small intestine. And then the fats are broken down into glycerol and fatty acids, and the fatty acids and glycerol end up getting absorbed into the lacteals, those lymphatic capillaries. So this is a nice review slide of the how material is absorbed in the intestine. Movements of the small intestine, we've already talked about these, but let's hit them again. You get the peristalsis, and that's just those wave-like pushing movements that push chyme down through the digestive system, through the small intestine. Segmentation are ring-like contractions, so you got a segment in the middle that's not contracting, and the segments on either side of that segment are contracting, and it squeezes the material together as a mixing movement. The parasympathetic nervous system sends out impulses that stimulate both of these, so peristalsis and segmentation. And then the sympathetic nervous system, remember, inhibits them. Sometimes you get if the small intestine becomes overstimulated or overly distended or irritated, you can get something called a strong peristaltic rush that sweeps all the contents into the large intestine, and that's how we get diarrhea, or one of the ways I should say. The ileocecal sphincter joins the ileum of the small intestine into the cecum of the large intestine. Okay, now on to the large intestine. Large intestine's named the large intestine because it's a larger diameter. It's only about 1.5 meters long. It opens at the distal end out to the body. There is some absorption that happens here, but mainly water and electrolytes. It reabsorbs a lot of the water that was used from the digestive process. And finally, it forms and stores that feces. The large intestine is made up of the cecum, this first part, the colon. And the colon has the ascending colon, the transverse colon, and the descending colon. And you've got the sigmoid colon, which looks like an S, hence the sigmoid. And then you've got the rectum and the anal canal. So that's like the anal canal is like the last 2.5 to 4 centimeters, and that opens the outside body. There's an internal and external anal sphincter, and the internal is involuntary and the external is voluntary. Right. Functions of the large intestine. Structural intestinal wall, the large intestinal wall. So same 4 layers as the rest of the alimentary canal, but the walls of the large intestine don't have that those villi like the small intestine does, because there's not as much absorption happening there, and there's the plica circularis either. There are these longitudinal muscles called tennicoli, and these tennicoli are 3 bands of longitudinal muscles that run the length of the colon, and the bands create these pouches called the haustra, and that's what forms the feces. So it's what helps make the feces in the large intestine. Okay, functions of the large intestine, like we said, it has little or no digestive function. Has those lots of goblet cells that secrete mucus, and that's really the only significant secretion in the large intestine. It absorbs about 90% of the water out of the feces and of the electrolytes. The other function with the large intestine is that intestinal flora, which we've mentioned before. That intestinal flora is just there. It's the normal flora, bacteria that live inside of us, and it breaks down things like our cellulose or our fiber. And intestinal flora also produces things like vitamin K and B12 and thiamine. And then the other function we already talked about, which was forms the feces and carries out defecation. Movements of the large intestine, just kind of similar to the small intestine, but they're slower, less frequent. You still have the mixing movements, and you get the peristalsis moving 2 to 3 times a day. For some people it's fewer, for some people it's more. All right, defecation reflex is relaxing the internal anal sphincter, and then we relax voluntarily the external anal sphincter. Feces is mostly water, there's some electrolytes in there, and mucus, bacteria, the bile pigments in there give it some color. The bacteria in our large intestine help to produce our, well, they produce lots of stuff, but the odor comes from the bacteria, and that things like ammonia are produced in there, phenols, indols, so all produce that characteristic smell. Here are some of the or table with a summary of all the enzymes for the digestive system. And then once again, lifespan changes, nothing but good news for everyone. Tooth enamel thins, gums recede, teeth loosen, just all good news as we get older. The digestive system, like all the other systems, starts to become less functional. One thing I do want to go back to on the large intestine was this appendix. And the appendix is attached to the cecum here. And one of the functions of the appendix now, sometimes one of the functions of the appendix is that when we do get waves of diarrhea, it forces a lot of that normal flora out of us. So the diarrhea pushes that normal flora out of us. And the appendix is a place where it's been found that that normal flora can regrow from, because we need that normal flora to live. Now, without it, we wouldn't be able to live. So when we have an episode of diarrhea where we wash a lot of that out, they can regrow from the appendix. Sometimes that normal flora in there causes the appendix to become inflamed and has to be removed, and that's when people have appendicitis. All right, that's it for the digestive system.